Hello, I'm Dr. Mike Darcy, and this is another episode of the Science on the Edge podcast. This is a bite-sized episode, relatively short, to give you an overview of a genetic engineering technique known as CRISPR. You may have heard of CRISPR on the news. It's touted as a technique that's going to allow the curing of a myriad of genetically inherited diseases, the genetic engineering of our species to increase lifespan, health span, reduce susceptibility to various diseases, and do all sorts of other miraculous things. First of all, is it true? Is it going to do all of these things? Well, in my opinion, probably yes. Probably yes. We've had genetic engineering techniques around for some time. Things like retroviruses have been used to genetically engineer our cells, and so have liposomes and various other techniques. Problem is, they're not very specific, they're not very accurate, and there can be potentially negative consequences to those techniques, which is why they have not really proven to be that effective in medicine or in research particularly. There's some useful outcomes from these techniques, but when it comes to general medicine and increasing the health and lifespan of our species, they have not really proven to be particularly useful. CRISPR, however, looks a bit different. And the reason for that is because CRISPR is incredibly accurate. It can target a specific gene and either delete that gene or edit that gene. So potentially an incredibly useful technique. So let's delve into exactly how it works. Okay, so we'll start with a general introduction, then we'll delve a little bit deeper. So first of all, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Palindrome is something that is read the same backwards or forwards. Don't need to go into the details of how that relates to CRISPR itself, and maybe I will in one of my deep dive episodes, but that is what it stands for. It was discovered in bacteria and acts as the adaptive immune system. So what is the adaptive immune system? Well, in humans, the adaptive immune system is the part of our immune system which responds to specific types of infection, responds to specific infections, identifies them, destroys them, and has a memory. It's different from innate immune system, which is a sort of general response to anything that doesn't look like it belongs in our cells. That's our adaptive immune system, but it turns out bacteria have an adaptive immune system as well, known as CRISPR. Now, CRISPR has allowed scientists to either delete or edit specific genes at specific positions on the genome without causing unintended negative consequences. With previous genetic engineering techniques, the problem was that you could damage other genes that were not your target. But CRISPR is far more specific. So let's explain how it works in the bacteria before we explain how we use it in the lab. So first of all, bacteria can become infected with viruses. These viruses are known as phages. Bacteriophages are viruses that infect a bacteria. So we have bacteria, and viruses, phages, can latch onto the surface of these bacteria and can then inject the DNA, the phage DNA, into the organism, into the bacteria. Now, normally that phage DNA would become integrated into the bacteria's genome, and it would carry the instructions to make more phages, more viruses. And the bacteria would basically become a virus factory, releasing the viruses that are created into the environment to infect other cells. So how does the bacteria defend itself? Well, it defends itself using a number of enzymes known as Cas enzymes. First of all, when a bacteria becomes infected with a virus, a complex known as Cas1, Cas2, these are two proteins linked together, they will search out any viral DNA that's floating around in the cell, and they will chop a piece of it out. They will chop a piece of that that viral DNA out, and then they will carry that viral DNA to the chromosome of the bacteria. They will deliberately carry this piece of viral DNA. It can no longer cause an infection. It's just a piece of viral DNA. They will carry it to the chromosome of the bacteria, and they will insert it in a specific region known as a spacer. Basically, there are a sequence of genes on the bacterial DNA that are together, some genes known as the Cas genes, followed by a number of spacers. Now, spacers are added by inserting viral DNA. So when the Cas1, Cas2 protein encounters a virus in the cell, it chops a piece of the DNA out and inserts it as a new spacer as part of the bacterial chromosome. So far, so good. At a later point, the cell might be infected by the same phage, the same virus. When that virus comes along, it will insert its DNA into the bacteria in an attempt to reproduce itself. However, when that occurs, we activate another protein, the Cas9 protein. So a Cas9 protein is activated. This protein is made from, it's basically transcribed from a sequence in the CRISPR complex, that region of the bacterial chromosome. The Cas9 protein is produced, and within it is inserted a spacer. Remember the spacer that contains a sequence equivalent to that of the virus, specifically RNA sequence. I'm not going to talk about the differences between DNA and RNA here, but let's just say if you have a DNA sequence and you make RNA from it, the RNA is the opposite sequence. The opposite and complementary, meaning RNA can bind to DNA, like a lock and a key. So as we have spacer DNA from a virus in the CRISPR-Cas9 region, and we can take that spacer, we can make RNA from it, insert the RNA into this Cas9 protein, and then the Cas9 protein will wander around the cell, and if it comes across any viral DNA, it will line up its piece of RNA with the DNA within the virus. And then an enzyme that is present in the Cas9 complex will snip that DNA. It will chop it up and therefore inactivate the virus. So just to summarize that one more time, when a bacteria becomes infected by a virus, viral DNA will try to insert itself everywhere to make copies of itself. However, we have an enzyme called Cas1, Cas2. This is a protein complex, which will look for any viral DNA that's just floating around in the cell. When it finds it, it will chop a piece out, and it will insert that piece into a region of a CRISPR complex, that region of the bacterial chromosome where the spacers are, and therefore insert a new spacer. The spacer will be a piece of viral DNA. Piece of viral DNA that cannot cause an infection, it's just a small piece. Then, if the cell is infected again, the Cas9 protein will be produced from the CRISPR complex. The Cas9 protein will have a piece of RNA inserted in it that is transcribed from the spacer, so a mirror image of the viral DNA. This Cas9 will then hunt out any DNA that matches the sequence of RNA within it, which is the viral DNA. And when it comes across any viral DNA, it will bind to it and it will chop that virus into pieces. Okay, so far, guys? Good. We are going to revisit this. What are the pros and cons of CRISPR? Well, let's start with some of those benefits. First of all, we can actually engineer CRISPR ourselves now. So rather than using viral DNA, we can basically create RNA sequences of any kind that we like and insert them into Cas9 complexes. So we can take Cas9 proteins and put an RNA sequence in that is the mirror image of any piece of DNA, including defective DNA within our cells. If we then allow the Cas9 complex into our cells, it will take that RNA sequence and it will match it to whatever gene we like. For example, the gene that causes cystic fibrosis. It will then line up with that gene and delete the faulty components, therefore perhaps resulting in the curing of the condition. So we can use the CRISPR system artificially to remove disease-causing alleles, so disease-causing genes from the genome. This could potentially increase lifespan by messing around with various genes and gene complexes, perhaps even increase intelligence, strength, and vigor in individuals. Perhaps remove negative alleles that left unattended would increase the person's chances of developing Alzheimer's, cancer, and diabetes, and all sorts of other diseases. We could remove those particular sequences in principle using this system. So lots of potential benefits. In terms of potentially harmful uses of CRISPR, well, it could be used to create genetically engineered bioweapons because it's a very effective way of engineering things such as bacteria. So we could use this for negative purposes, but we can sort of do that now anyway. But perhaps there could be bioweapons or such created using CRISPR. There could be negative consequences to health or fertility if CRISPR is used in a widespread way in the population. Because if we do use it in a widespread way to edit many, many genes in the human genome, well, we don't know how every pathway in our cells interacts with every other pathway. So there could be unforeseen long-term problems because of the genetic engineering, which could detrimentally affect our species. Creation of designer babies. Obviously, with the ability to edit genes in very specific loci to change all sorts of traits in an organism, well, this technique, if used in, for example, IVF clinics, could result in the creation of designer babies. You want your baby to have this trait removed, this trait removed, this trait added, this trait edited, then that is becoming increasingly more easy to do with CRISPR. So designer babies could be a consequence. Whether you believe that to be a negative consequence or positive consequence is really a matter of perspective. Ultimately, we could end up with a separation of humanity into the genetically edited and the non-edited. These techniques might cost money. They might cost a lot of money. So you may end up with people in richer countries or richer members of poorer countries having their children engineered, having their own bodies engineered to become stronger, more intelligent, more longer lived, etc. And these changes might be passed on to the next generation as well. So you end up with almost a species of supermen and superwomen. Meanwhile, the people that can't afford it, well, they stay normal. At the moment, you see, doesn't matter how poor you are, poor person's brain, poor person's genetics is not really any different than a rich person's brain and a rich person's genetics. So poor people can rise to the top. But if the people at the top are in effect a different species, that might be very difficult to accomplish in the future. So we may end up with 2 species or more on this planet because of techniques like CRISPR. So positives and negatives. Okay, let's talk more about the genetics now. So here are the Cas genes. This is what the region of the bacterial genome looks like where CRISPR is made, where the process is set off. So we have these Cas genes such as Cas1 and Cas2 and Cas9. And then next to them, we have these spacers, these little spacers where the viral DNA from repeated infections is inserted. You can end up with many of these spacers. It's a history of the infections that the cell has been exposed to. So here's the complex. There's a Cas9 protein. Now the Cas9 protein has cRNA inserted in it. What's cRNA? Well, this is basically the piece of RNA that's generated from the virus. So basically, you take that little bit of DNA that's in the spacer, you make some RNA from it, you actually link that to something called the tracer. Tracer RNA, this just allows the Cas9 protein to actually bind to that RNA that's been made by the spacer. So you get your piece of cRNA, which is basically made from the spacer. It sticks to a tracer, and this allows the Cas9 protein to bind. It binds by holding on to that tracer, basically. And now you have a complex, the Cas9 complex, which has got the tracer within it and the cRNA, this sequence that is specific for a virus, or for whatever DNA you want. Originally in nature, it was designed to attack viruses, but we can use it to attack any DNA we want and edit that DNA, even within a human genome. Okay, so once you've got this complex, it will seek out the DNA, whether that's viral DNA or DNA in some organism that you want to genetically engineer. It will bind, and when it binds, its RNA will line up with the DNA sequence that it's specific for, and enzymes within the Cas9 protein will snip that DNA out. Okay, so let's tie that together. So here is the whole thing. I've given you the information in little bits, and I've left a few bits out at one section and put in some different bits in another section. Well, here it's all gonna come together, and hopefully it will make some sense. So DNA from some sort of phage, some virus infects the cell. It's processed into short segments that are inserted as spacers into the CRISPR sequence. CRISPR repeats, and spacers in the bacterial DNA are transcribed into RNA. This RNA chain is cut into short pieces called CRISPR RNAs. So CRISPR RNAs. As CRISPR RNA sequences are copied from the viral DNA sequence, they are exact matches to the viral genome and thus serve as guides. Scientists can design any guide RNA to match any gene. So for those of you watching this on YouTube, rather than listening to it on a podcast app, there's an image here that shows the whole process. The process basically involves a piece of tracer RNA being produced from part of the CRISPR complex. This tracer RNA is made. The Cas9 gene is also transcribed, making the Cas9 protein. And spacers are actually produced as well as part of a pre-cRNA complex. And then the whole thing is tied together. So the Cas9, first of all, the tracer RNA will bind to the pre-cRNA. It will bind to it, and this will guide the Cas9 protein to the complex. So the Cas9 protein will now bind to the piece of pre-cRNA, which was generated from the spacers, and it will attach via the tracer RNA, making a complex. At which point, any parts of the pre-cRNA that are not needed, the overhangs, will be chopped out, leaving a completed Cas9 protein that is ready to go and find a specific gene and to edit it. Okay, so what are my thoughts on this technique? Well, this technique, when used in animals, in plants, even in humans, holds huge potential. We could remove so many negative alleles from our genome, get rid of so many defective genes that cause disease or reduce lifespan. Already this technique is being used to cure, in effect, cystic fibrosis, sickle cell disease, and Huntington's disease. These are diseases which do not have a cure at the moment and will reduce lifespan substantially. But CRISPR could get rid of all of these diseases fairly easily, actually, and fairly soon. That's where this process will begin. But then we'll start, once we've got rid of genetically inherited diseases, we'll start looking at more minor gene defects, so defects which just reduce lifespan by a year or two, or an increased likelihood of developing diabetes or Alzheimer's or heart disease or stroke or arthritis. And we'll start removing those to make a species that is more robust, tougher, stronger, more long-lived. This technique, with a slight modification, can actually be used to insert new genes into the genome. You chop out a defective gene, but you also insert a replacement gene using a modified version of this technique. So you can actually remove a faulty gene and insert a good gene. This together means that we could completely re-engineer our species. Personally, if this is done right, I think it will be an amazing, amazing thing. Our species will be stronger, more long-lived, more healthy, more intelligent, all of these things than it's ever been. I'm optimistic about this. However, I'm also aware that this could be misused. It could be misused massively. And CRISPR could be used to actually create designer babies in a negative way, to perform a kind of eugenics even. So there are many negatives, and some of the negatives might be unforeseen. We don't know what playing around with many of our 20,000 genes in different combinations of ways might be. We don't know what the long-term effects might be to our species as a whole. We may regret the whole enterprise. Time will tell. Okay, well, that's all for now. If you want to know more about me, then you can follow me on Twitter at Mark of the D. That's at MARKOFTHE capital D, or you can visit my website, which is scienceontheedge.com. And if you'd like to support my podcast, then you can make a donation via the donate button on scienceontheedge.com. I'll say that one more time, at scienceontheedge.com. Just click the donate button if you would like to support this podcast.